This is part 5A for the microbiology class. There are not very many animations in this section simply because they were not available at the time of recording. What exactly is clean? In microbiology, there are specific differences in the terms one might use to call something clean. One may be used to describe something as clean or not having any microbes, and sterilization is going to be the term that's used to refer to the removal of all microbes. This includes viruses and endospores, but does not apply to prions. Aseptic is an environment free of contamination by pathogens. Disinfection or use of disinfectant is the use of physical or chemical agents to inhibit or destroy microbes. An antiseptic, or the process of asepsis, is the use of chemicals used on the skin for disinfection. Degerming is the removal of microbes from a surface by scrubbing, such as hand washing. Soap is used to assist in loosening the organisms. Sanitation is the process of disinfection of places or utensils used by the public, decreasing the microbes for a public health standard. Pasteurization uses heat to kill pathogens and decreases microbes causing spoilage. Many of these kill some microbes, but not all. Something that is static inhibits growth or metabolism, but does not necessarily kill the organisms, while an agent that is cytal destroys or permanently inactivates a particular microbe. The microbial death rate is the permanent loss of reproductive ability under ideal environmental conditions. This is described as a constant over time for any particular organism under particular conditions, such as 100 organisms per hour. There are a few ways antimicrobial agents work. One way is by altering cell walls. The cell wall counteracts osmosis when in a hypotonic solution, so a cell without a cell wall will burst. Damage to the cytoplasmic membrane also allows the contents to leak, killing the cell. Another method is to damage proteins or nucleic acids. Proteins help regulate cell function, and denatured proteins do not function, causing cell death. Nucleic acids can be altered or destroyed, resulting in fatal mutations. The efficacy, or how effective an antimicrobial is, depends on several things. One concern is the site being treated. Agents must not be too harsh or extreme when used on humans or animals. It must not be too harsh to damage fragile objects. Some instruments that penetrate the body and its defenses must be sterilized. Susceptibility from most susceptible to most resistant is going to be in the following order: enveloped viruses, gram-positive bacteria, fungi, gram-negative bacteria, active stage protozoa, cysts of protozoa, mycobacteria, and bacterial endospores. Most bacterial endospores can live in the soil and are virtually impossible to destroy using any practical means. High-level germicides kill all pathogens, including the endospores. This is used for cleaning invasive instruments. Intermediate-level germicides kill fungal spores, cysts, viruses, and pathogenic bacteria, but not endospores. These are used for instruments coming in contact with mucous membranes, but are not adequate for invasive instruments. Low-level germicides kill gram-negative bacteria, fungi, protozoa, and some viruses. These are used for items that only come in contact with the skin. The environmental temperature and pH influence the effectiveness. Disinfectants tend to work better at warmer temperatures rather than cool. Some are more effective at lower pH values. Some materials may interfere with disinfection, so they must be cleaned before sterilization. Even items in the autoclave must be washed beforehand. There are tests that may be done to evaluate the effectiveness of disinfectants and antiseptics. The phenyl coefficient compares the ability to control microbes to phenyl under standardized conditions. A ratio greater than 1 indicates the agent is more effective than phenyl, while less than 1 is less effective than phenyl. The greater the ratio, the more effective. This comparison is no longer used. With the use dilution method, metal cylinders are dipped into broth cultures and dried at 37 degrees Celsius. Each cylinder is immersed in a different dilution of disinfectant being evaluated. After 10 minutes, it is removed, rinsed, and dipped in fresh sterile media for hours. The most effective is the one that entirely prevents growth at the highest dilution. This is the current standard of evaluation. The disc diffusion, or Kirby-Bauer method, uses paper discs soaked with an antimicrobial agent of standardized concentration. The disc is placed on a solid medium which has been heavily inoculated. These samples are incubated overnight and observed for a zone of inhibition around the disc. In general, a larger zone is more effective, but may only indicate how easily the substance diffuses in agar. A standard table may be used for comparison. The in-use test compares the swabs taken from an actual object before and after the application of disinfectant. First, a swab is taken from the dirty object and inoculated on appropriate media. The new swab is taken after the object had the disinfectant applied and inoculated on the appropriate media. Both are incubated and examined for growth. There are also physical methods to control microbial growth. Several of these involve altering the temperature. The thermal death point is the lowest temperature that kills all cells in a broth in 10 minutes. The thermal death time is the time amount to completely sterilize a particular volume at a set temperature. Moist heat kills by denaturing proteins. Water is a better conductor of heat than air. You have experienced this by being able to reach into a hot oven, heated up to 350 degrees, but not able to put your hand in boiling water at 212 degrees Fahrenheit. Boiling kills vegetative bacteria, fungi, protozoa and trophozoites, and most viruses within 10 minutes at sea level. The autoclave uses a chamber to add pressure to heat. The steam gets to 121 degrees Celsius at 15 pounds per square inch. This destroys microbes in small volumes in about 10 minutes, although it is usually done for 15 minutes for added safety. More time is required for larger volumes. Pasteurization does not kill all organisms. Heat-tolerant prokaryotes survive, but they do not cause spoilage. The time and temperature vary with the product. Milk requires 72 degrees Celsius for 15 seconds. Dry heat is used for things that cannot be exposed to steam, such as flaming the tip of the tube or flask in the lab. This requires a higher temperature over a longer time. It also works by denaturing proteins and oxidizing metabolites. Refrigeration or freezing decreases microbial metabolism, growth, and reproduction. The psychrophiles can multiply in the refrigerator and spoil food. Ice crystals can puncture cell membranes in a slow freeze, but it does not kill all microbes. Desiccation or drying inhibits growth because of the metabolism requiring water. This does not kill all microbes, and molds can grow on as little as 16% water. Lyophilization combines freezing and drying. This can preserve microbes for years. They are instantly frozen in liquid nitrogen or dry ice, and water is vacuumed out. High concentrations of salt or sugar in food can be used to alter the osmotic pressure and inhibit microbial growth. Filtration can trap microbes larger than the pores of the filter. Membrane filters made of nitrocellulose have a pore sizing from 25 microns to less than 0.01 microns, so we are now able to filter viruses. At the time of recording this video, it is still not practical to use filtration for outdoors equipment. One of the limitations is the filters don't have any way of letting us know when they're no longer being effective. Prions are able to pass through holes of 0.01 microns. Radiation is useful in destroying microbes. Electromagnetic radiation, with its shorter wavelength, is more suitable because it carries more energy and is better penetrating. Ionizing radiation from electron beams, gamma rays, and X-rays has a wavelength shorter than 1 nanometer. It disrupts hydrogen bonding, oxidizing double bonds, and forms a reactive hydroxyl. It is used on lab equipment because it cannot penetrate deep into matter, and is only used for thin foods. Non-ionizing radiation with wavelengths greater than 1 nanometer include UV light, visible light, infrared, and radio waves. Of these, only UV light has enough energy to be useful. It does not penetrate well, so it can only be used for transparent fluid, air, and surfaces. Many chemicals are used to control microbes. Phenyl denatures proteins and disrupts cell membranes. It is considered intermediate to low-level disinfectant and is used in home and healthcare settings. An example is Lysol. A disadvantage to phenyl is the bad odor, and it may irritate the skin. Alcohols are intermediate-level disinfectants. They are not effective against fungal or bacterial spores. Isopropyl and ethanol denature proteins and disrupt cytoplasmic membranes. They require the presence of some water and are used in concentrations of 70 to 90% alcohol. The halogens include iodine, chlorine, bromine, and fluorine. These are intermediate disinfectants that work by denaturing or oxidizing enzymes. They are effective against fungal spores, some bacterial spores, and protozoan cysts. Iodine is used as an antiseptic, chlorine in bleach, and bromine in hot tubs. Chlorine is also used in swimming pools in some cases. The oxidizing agents are substances such as peroxides, ozone, and peracetic acid. They work by oxidizing enzymes and are used in high-level disinfectants, which are good for inanimate objects. They are not good for wounds because human cells have catalase that neutralizes it. Ozone is used for drinking water and peracetic acid for surfaces. Surfactants decrease the surface tension of water and include soaps and detergents. Soaps are good degerming agents to loosen substances from the surface, but are not antimicrobial. Detergents have positive charged surfactants that are used to loosen and are antimicrobial. The quaternary ammonia compounds are used in industrial and medical applications because they are harmless to humans. They are found in mouthwash and work by disrupting cellular membranes. Because Pseudomonas grows in them, they are considered low-level disinfectants. They are not effective against non-enveloped viruses, mycobacteria, or endospores, and they are deactivated by soaps. The heavy metals include arsenic, zinc, mercury, silver, and copper. They combine with sulfur and cysteine to alter the three-dimensional shape of proteins. Silver nitrate is used in the eyes of newborns to kill potential Neisseria gonorrhea causing blindness. Thimerosal contains mercury and is used to preserve vaccines, although the use is now discouraged as of 1999. Copper interferes with chlorophyll and is used in algae control. The metals copper, zinc, and mercury are used to control mildew in paint. The aldehydes include formaldehyde and glutaraldehyde. They work by cross-linking amino, hydroxyl, and sulfhydryl and carboxyl groups. Medical and dental equipment is disinfected with a solution of 2% glutaraldehyde. Formalin for embalming or disinfection of inanimate objects is made with 37% formaldehyde. Formaldehyde is carcinogenic. Gases such as ethylene oxide, propylene oxide, and beta-propiolactone are used to sterilize objects that can't use heat or chemicals. They work by denaturing proteins in DNA. Gases are very poisonous and are potential carcinogens and must be used with great care in special facilities.